Okay, so where we left off was on the cytoskeleton, which is a fiber network inside of the cytoplasm. What it helps to do is organize things and also keep things from moving into the wrong place. It helps in some of the functions as well. So it's going to provide some mechanical support for shape. Some of them actually provide motility in there, so your cytoskeleton is going to be made of the same stuff as cilia and flagella and connected to those. It provides transport pathways inside the cell, so you're not just sending things out randomly hoping they get to another part of the cell. There's actually a pathway that it can follow in there. It can help to move the inside of the cell when it wants to do phagocytosis. And then it also helps in regulating some of the biochemical activities because of the way that it can guide things to go into different places in the cell. And so this is an example here in this picture showing where you've got your microtubule and you've got a vesicle that actually has these little motor proteins that walk it along the pathway in there. And then just an electron microscope picture of it. So the cytoskeleton is going to have a few different types of fibers in there. The microtubules are the thickest of them, and they're made of a protein called tubulin. They help to provide the shape of the cell. In there, they're going to provide tracks for the organelles to move along and help to guide the secretory vesicles. In animal cells, we've got a structure called the centrosome, which is a region that the microtubules will grow out of. It helps when the cell divides in there as well. The centriole is within the centrosome, and it's going to have these 9 sets of triplet microtubules in a ring. And this one is going to be the exact part of it that is involved in animal cell replication. We know that it's not 100% tied to just replication because you do have plant cells and yeast cells that don't have those. So this shows how you've got these 9 triplets that are in a ring here. And then this is just a cross-section of it on an electron microscope picture. So the cilia and flagella are going to be microtubule-containing extensions. They're made from microtubules, and they're going to project from the cell so they can use it for motion in there. Flagella do more of an undulating motion, which is like swimming. The most common example that most people have probably seen on TV or in videos is sperm swimming, like a fish. And then the cilia are going to work more like oars, and so they have a power and recovery stroke, so they do more of the pulling in there. So it almost looks more like crawling like a spider in there with the cilia. So the cilia move at a right angle to it, so if you're going like this, you're going this direction with the cilia, where the tail for the sperm is just behind in there, and they kind of just, it almost looks like a little snake moving in there. So primary cilia can act as a signal receiving antenna and transmit molecular signals. And so these are something that we've found, and they appear to be crucial in brain function and some of the embryological development in there. So they actually kind of work like a receptor to get messages in there. So the pattern of them is going to be, depending on which thing you're looking at, the 9 plus 2 means you've got 9 doublets of microtubules arranged in a ring, and then you've got 2 in the middle in there, 2 singles in there. So this is what you're going to see in almost all of the eukaryotic flagella and the cilia. And this is just showing the 9 plus 2 in there. The 9 plus 0 is going to be what you see, 9 doubles of tubules in there, but nothing in the middle. It lacks those central pair. That's what you're going to see in the non-modal primary cilia in there. So they'll be a little bit different. So where the microtubules attach is going to be a basal body that helps to anchor it in there. So the proteins that are going to protrude along are called dinins, and these are going to be along the length of the cilia and the flagella. They're going to be what we call a motor protein in there that changes shape with ATP. So it will be straight in one position, bent in the other, so it can bend back and forth like this. And that's what actually allows it to have the motion in there. So this is kind of showing it being bent and straightening up in here in this image. And this is showing it going back and forth to make the wave-like motion. So the microfilaments are going to be a little bit smaller in there. These are solid rods that are made of a protein called actin. Actin is also present in muscles in there. And it's a more globular protein. So things these are going to do, they're going to help to support the cell shape as well. They're going to help give a cortex and outer cytoplasmic layer in there to give it more of a gel consistency. It will help to bear tension forces. So if you're going to do phagocytosis, you're going to have the microtubules that move the cell to be able to do that. But this is going to also help to reinforce the cell, so that when you do that, you don't just have a piece of the cell that gets lobbed off in that stretching process. It's going to make it a little bit stronger in there. So they have motility. They've got a contractile apparatus in there with, and you look in the muscle fibers that they'll work with the protein myosin in there. And this allows the amoeboid movement in there. So it also is going to be responsible for cytoplasmic streaming, which is a circular flow of the cytoplasm within the cells. It's just going to keep the cytoplasm moving around in there so it's not sitting in one place. Cells don't have a circulatory system, but they do need to keep the fluid moving in there to keep nutrients and things like that moving around. So that's what this will help to do. So this picture just shows them some actin filaments in here in a muscle cell. And it also shows how you've got some of them inside of another cell here. And this is showing kind of acting for cytoplasmic streaming to move stuff within that plant cell. So then we have intermediate filaments. So your cytoskeleton has your intermediate filaments, microtubules, and microfilaments. There's 3 different parts to it. So these are going to be a little bit larger than the microfilaments. These are the middle-sized one. They're specialized to bear tension in there, and these will persist after the cell dies. And they're going to make up the nuclear lamina that lines the inside of the nuclear envelope. So it's just one more level of reinforcement and strengthening things inside the cell. So extracellular components, these are things that are outside the cell. You've got the cell wall, and that's going to be extracellular in plants. Also, some bacteria will have cell walls. Animal cells do not have cell walls. That's one of the big differences in there. One of the things by having this harder cell wall, it helps to hold the plant up against gravity. So plants can get pretty tall and they don't have any bones in there to hold them upright, but the cell walls provide some of that support in there. So you'll see them in the prokaryotes, which are your bacteria. Cell walls are going to be in a lot of fungi, and some of your protists will have cell walls. So a cell wall is thicker than a plasma membrane, and you're going to have microfibrils in there that are made of cellulose in there that are going to help to embed in the matrix, along with other polysaccharides and proteins in there. So different species and cell types will have different types of polysaccharides and proteins in there. So these are going to be nice strong fibers in there in that matrix. So that cellulose that you can't digest in plants is what helps give them that strength for the cell wall. So it needs to be tough stuff in there. And that's part of the reason we don't digest it is we don't have the enzymes, but you also want them to be tough, and to have an enzyme that could do that would probably damage other things because of the strength of these. So the primary cell wall is what you're going to have in a young plant cell. It's first secreted as a relatively thin and flexible cell wall. And then the microtubules in the cortex are going to guide cellulose synthase and help to orient cellulose deposits in there to help build the cell wall a little bit more. Then you've got the middle lamellae, which is going to be between the primary walls of the adjacent cells. And it's basically sticky polysaccharide in there, so things like pectins and glues. So if you get pectin, like you want to make something with jam in there, fruit pectin in there, to help make the jelly sticky and hold together, this is where pectin is derived from. And that sticky property in there is what the plant cell is using it for. And you use it also to make things sticky. And then you've got the secondary cell wall that's going to be between the plasma membrane and the primary cell wall in a mature cell. So some cells will just harden the primary cell wall, some will make the secondary like that. And this is just showing the different layers in there in a cell wall. So sometimes we want to be able to have the cells be able to talk to their neighbors in there. And so the plasmodesmata are perforated channels between the cells. This is pretty common to see in plants. So that way when one cell has nutrients, it can actually pass it on to neighboring cells. And we actually see a similar thing like in bone cells in humans to be able to pass nutrients on. So this is just showing where you've got these little channels, the plasmodesmata, in there between the cells. So the extracellular matrix, this is going to be stuff outside the cells in animal cells. You've got glycoprotein, which the most common one is collagen. So it's just made up of the combination of sugar and protein in there. This is going to form really strong fibers. So in terms of making something synthetic that would have similar strength to a size ratio, we don't have anything we can make synthetically that for the size has the same strength as collagen. It is really strong stuff. So that's the reason why you don't have bones sticking onto your ears here. That's the reason they stay onto your head and don't just fall off. They're just held on there with cartilage, or not cartilage, but the collagen in there is what gives them the strength like that. Now, yeah, you can't tear a human ear off, but that's when you look the amount of fibers in there and the strength you get from it, it's pretty amazing. It's kind of like spiderweb silk, where you see this thin strand and you go, how does that big spider hang from that? It's just a really strong material for its size. So the proteoglycans are small core proteins that are going to have carbohydrate chains in there. It helps to form a network around where you have the collagen. So it's just one more piece of that matrix in there. Fibronectin is going to bind to the surface cell proteins called the integrins in there. The integrins are going to actually span the plasma membrane in there. So in this picture you can see you've got your plasma membrane here, cytoplasm on the inside. So you've got your integrins, which are these proteins spanning the membrane. And then you've got the collagen fibers in here. And then the proteoglycan complex that's helping to kind of hold that in place in there. And then the fibronectin that helps to connect it to the integrins. So does that make sense when you look at the picture of it? I know otherwise they just look like a bunch of names of things that you go, they do what? So when we have cells close together, we want to be able to have some intercellular junctions. So the plasmodesmata are perforating channels in the cell walls where the cytoplasm can or the cytosol can actually pass through and chemically join the cells to be able to pass the nutrients in there. What we'll see in animal cells are going to be these tight pressed, tight junctions in there. And so they'll be bound by specific proteins, but it's going to help to hold the animal cells together. So this is why your skin can be watertight in there, because the cells aren't just sitting next to each other. It's like they're glued together in there. So the desmosomes are going to attach muscle cells together. They're going to be made up of intermediate filaments of keratin. And then you've got gap junctions that are going to be between cells for communicating, so places like the heart and muscle cell. So different types of cells get different names for their little openings in there, but in a multicellular organism, the cells are usually going to be joined together, and they're usually going to have some form of communicating between the cells to be able to pass the material through. So cells are a living unit greater than the sum of its parts. There is something about having all of the parts together, working together in a living unit, that they will have some different properties than if you were to just take the parts by themselves and look at them as if they weren't alive. So with 7, this is the stuff that is in there that you can go through on your own. If you have any questions, don't hesitate to ask on there. It's a relatively short section that is relatively easy, which is why I chose 7 to be the section you'll go through on your own in there. When we have labs on Wednesday, if you have questions on any of these things, we can go through them, or if you have questions before that, we can certainly set up a time and I can go through the stuff with you. So in Unit 8, this is where we will start to go into metabolism. So our next section will be, for your next test, a lot of the metabolism stuff. That's probably the hardest section for people in there, is the metabolism, just because there's so many chemical names to it. The good news is you will go over metabolism just about in every biology class you take. So eventually, when you hear this stuff enough, it does start to sink in a little bit more in there. So metabolism is going to transform between matter and energy. Because you're going between matter and energy, it is going to be subject to the laws of thermodynamics. So if it's a chemical reaction in a living system, we call it metabolism. So we organize it into metabolic pathways. And a pathway is just going to begin with a specific molecule that will go through a series of steps to be altered, and you end up with something else at the end. Some of the pathways are going to be catabolic in that they break things down. Others will be anabolic, that they're going to build things. When you break things down, you're going to release energy. So if you think of it like when you break down leaves, if they're decomposing, it releases some energy. If you burn a tree, it releases some energy. In order to build something, you have to put energy in. So if you're going to grow a plant, you've got to put light energy into it to do that. So bioenergetics is just the study of how energy flows through living organisms. So forms of energy. Energy is just the capacity to cause change. So we can have kinetic energy, which is the energy of motion. And there, the more something moves, the more energy it has. Heat or thermal energy, this is actually created by the kinetic energy associated with the atoms moving in there. So when you heat something up, what you do is just make the atoms move faster in there. So they've got more energy, and you experience it as heat. So potential energy is energy due to location and structure. So chemical energy is the potential energy that can be released in a chemical reaction. So in its structure, in there, in chemical reaction, you've got bonds holding something together. That chemical energy is the potential energy you get when you break those bonds in there. Fusion actually requires energy to put things together. And it takes a lot of energy. That's been the big problem with it, is how can we create enough energy to drive things together and control it without blowing everything apart. Yeah, right now the only way we know to create that much energy reliably is through nuclear fission, splitting something apart, which starts a chain reaction that we really don't want to have to clean up just to make a couple things go together. So this picture here just kind of demonstrates it in here, that this person climbing out of the water is using up some kinetic energy to try and get out of the water. So they've got movement, but they're also moving up higher. This person has more potential energy because they can fall down and do some movement. So this person is doing the kinetic energy because they are actually doing the falling. Well, they are up here, it's potential energy. Well, it's actually falling. Now you've got the kinetic energy. And this guy or these people in the water have the least energy, and they're there at the bottom, and they're going to have to put in some energy to get out and have some potential energy again. Yeah, this guy's not burning a whole lot of calories there. So light is also a form of energy. It's a little bit, light's one of those things that's a little bit weird because it also can behave as a particle in there. So it's a little different for understanding light in there. So thermodynamics is the study of energy transformation. And when we look at a system, you are studying everything that can interact with the matter in there. So in an isolated system, it's unable to exchange matter or energy with the outside surroundings. It's just completely isolated, doesn't interact with anything else. With an open system, matter and energy can transfer between the system and its surroundings. So which one of those do you think is the more common one to see? An open or isolated system? Open. It's actually really difficult to create an isolated system in the lab. So you're going to operate as an open system. Whether you like it or not, you're going to interact with your surroundings. So when you go to do metabolism in your body, it's going to be affected by your surroundings in there, by the temperature. If you heat up the temperature or if you decrease the temperature, it will change how your metabolism reacts to that. So the first law of thermodynamics says energy cannot be created nor destroyed, but it can be transferred and transformed. It's also called conservation of energy. So if you are releasing energy from something, it has to go somewhere. It doesn't just go into oblivion, never to be seen again. And it's got to transfer from one thing to another. So if you are going to break down something, that energy from breaking it down has to go somewhere. So in your body, what you're going to do is, if you break down food, you're going to harness that energy in something called ATP and then use it to fuel something else. So the second law of thermodynamics says during the transfer or transformation, some energy becomes unstable. Usually it's heat in there, and heat makes the universe more disordered. So when you go between things where energy isn't created or destroyed, something that would be 100% efficient would be able to take 100% of the energy you get from this and move it and end up with 100% over here. That doesn't happen, even though that first law of thermodynamics says it will. There is some along the way that we just can't harness in there that's going to become heat. And even though we can't necessarily gain that heat back, how that heat goes into the universe is it creates more disorder in the universe in there. So that's our explanation of it's not gone, we just keep increasing the disorder in the universe. So entropy is that measure of disorder or randomness in there. So every energy transfer or transformation is going to increase the entropy of the universe in there. So you may look at that and go, well, okay, if we never get this back to decrease the entropy, at some point are we going to just put so much disorder and randomness into the universe that it's going to blow up? We don't know. We've never had a universe other than this one that we have lived in to have any knowledge on that. So we don't really know how much it can hold, how much it's going to go. We just know that that accounts for where that energy goes. But what is going to happen in the long run, none of us really know. So increased entropy is evident in the physical disintegration of systems with an organized structure. So things eventually break down in there. So anything that you are using to harness energy, it eventually just kind of wears out and breaks down in there. So it's a spontaneous process, or for a spontaneous process to occur without putting energy in, it's going to have to increase the entropy in the universe. So for a process to happen spontaneously, it has to go from a higher energy state to a lower energy state. Things always want to move to the lowest energy state. So if you look and you set a ball on a hill, it automatically wants to move down to the lowest point. If you set water on a surface and it's got the opportunity to roll down to the lowest point, it will in there. So when those things happen, though, that change in energy becomes entropy. So things are not going to spontaneously roll uphill. They'll spontaneously roll downhill in there. So that's why we say for something to spontaneously occur without putting in energy, it's going to have to release some, and it's always going to lose some as entropy in there. So does that make sense? That idea that things don't just, it's like you don't spontaneously build muscles without putting in some work to get them in your body. So living systems increase entropy of the surroundings, and it takes more ordered forms of the surroundings and replaces it with less ordered forms in there. So the free energy is going to be the change in energy in the reaction that will determine if it occurs spontaneously. So we don't normally look at reactions and say, okay, I want to be able to measure the entropy. We don't really have numbers for that, but what we do have are numbers for free energy in there, and it's called the Gibbs free energy, and the way it's represented is as this delta G. So I know on some of the things, if it's the font for the delta doesn't always transfer into Canvas when it uses the whatever it does to convert Microsoft Office documents in there. So you may see it say delta, or you may see it say DG if you're reading it directly on campus. If you print it out as a Word document, then it's all happy with the little delta symbol in there. But just be aware that if you see something weird for an abbreviation there that doesn't make sense, it's that whole font conversion thing that's behind it. So the Gibbs free energy is the portion of a system's energy that can perform work when the temperature and pressure are uniform in a system. So we have this equation, delta G is delta H, the change in the system's enthalpy, minus the absolute temperature in Kelvin times the system's entropy in there. This is what is behind it. What you will use more in an equation in chemistry would be this equation here. This delta G is the G at the final state minus G at the initial state. So it's kind of like saying if you want to know how much your dog weighs and they don't like to stand on your home scale, you stand on the scale with your dog, then you put the dog down and you stand on the scale by yourself and subtract the difference to find out how much the dog weighs. That's what this is doing to find your delta G, looking at the amount at the end minus the amount at the start. So when you do this, delta G is only negative when there is a loss in free energy in there. So for something to be spontaneous, do you think it would want to have a positive delta G or a negative delta G? So for something to be spontaneous, when you look at that equation, would you want it to have a positive or a negative delta G? Negative. Yeah, because you're starting out with your final state and you lose something from the final state. So the original state that you subtract from it was something a little bit more in there that you've gotten rid of. I know what you're saying in there. So the negative is what you're going to see for something being spontaneous. That's how it comes about to be that negative in there. Because I know sometimes when you look and you go, negative number in here for stuff like that, it can be kind of overwhelming. But this is, so if you are one of those people that needs to figure out the math behind it to know why, there's your equation to do it. If you can accept it as it is, then do that too. So as the reaction is going to proceed towards equilibrium, the free energy of the mix of the reactants and products decreases. And if it moves away from equilibrium, it's going to increase in there. So where things want to do is they want to always fall down and get delta G to 0, is what nature wants to do, is just bring that back to 0 in there. So an exergonic reaction is going to be one that proceeds releasing energy in there. The way I remember it is exergonic, think of it as the energy exits on there. So it's going to have a negative delta G, and the magnitude or size of the delta G is going to represent the maximum amount of work a reaction can perform. So for cellular respiration, so if you take glucose from your body and you combine it with oxygen, and what you're going to end up producing is 6 carbon dioxides and 6 waters. This is going to happen spontaneously to break this down, and the negative delta G for that is negative 686 in there. So you get 686 calories in there. 2870 kilojoules per mole in there. So if each mole of glucose is 180 grams broken down under standard conditions, which is going to be 1 molar concentration, 25 degrees Celsius at a pH of 7, you have 686 kilocalories available for work. So the products have 686 kilocalories per mole less energy than the reactants do. So when you were to break that down in there, what you're going to do is take that energy difference and you're going to store it as ATP in your body. You use that energy difference that you have from taking that glucose and breaking it down to make your muscles work or other things in your body work. Looking at what the starting thing was after what the final was, after you've, that's why it's negative. If they wrote the equation the other way around, it would be different, but they didn't. So an endergonic reaction is going to be one that has to absorb energy from the environment. Endo means in. It means you have to put energy into the reaction to get it to take place in there. So it would have a positive delta G in there, so it's not going to be spontaneous because energy has to be put in. So this little diagram here is kind of nice. This is your exergonic reaction. So you've got your reactants that you start out with and the amount of energy they have. The products are lower. The delta G represents this difference in there. So here the delta G is less than 0. If you look at an endergonic reaction, you've got your reactants here that you start out with, your products that you finish up with. This is how much energy you had to put into it to get it up here and there. So that one has a positive delta G. So I don't believe I put any of those numbers on the test. If you want to know the delta G of something, there are tables that you can easily look it up. That in chemistry, that's what you use. You go, okay, is this going to be spontaneous and how much energy is going to come from it? You use these tables for it. I don't really see any purpose in memorizing this number here. It's there so you can have a relative idea. But this is in laboratory conditions in here. Once you're actually in the body, it's going to be different than that, and you'll get different numbers. So I wouldn't worry about memorizing that number. There is just lots of different chemical reactions that occur. And I did see one chart that it was like this poster you could buy that had all of the biochemical reactions. And it had like 8-point font, and it took up the whole wall in there. So there is a chemical reaction behind everything that goes on in the body that's going to have its own delta G associated with it. But there's no quick, easy way of memorizing all of those. It's more of, what usually ends up happening is you're getting an overview in here of that these things all exist. If you become really specialized in an area of study, you'll know those sets of reactions. Otherwise, the rest, you know they're out there, and being able to rattle them off the top of your head would do you no good. It would serve no purpose. So does that kind of make sense in there? So with equilibrium and metabolism, so in a lab, reactions tend to like to come to equilibrium, where it's kind of like you go, okay, I'm going to do this, and it goes too far this way and just comes back and settles in the middle, like one of those little balls that goes back and forth in there. That doesn't generally happen in a living system, though, because usually if you're making a product in there, that product has a job to do somewhere else. So when the ball is swinging back the other way, somebody's coming along and taking that product away. So your body never looks and says, oh, I'm building up this pile. That pile never builds up. Somebody else is using it before it happens. So they never reach equilibrium. So your reactions are usually pretty much constantly being drove in one direction like that. So how we're going to harness the energy in the body and in most living systems is through a molecule called ATP in there. It's kind of like an energy currency. So when you break down glucose in there in your bloodstream, you need that energy from it other places in your body, not just in your bloodstream. So if you can create it as ATP, you can move that energy around and transfer it. So it's kind of like if you go to work someplace, maybe you made candles at work. They could pay you and saying, all right, you've made a lot of candles for us, you can take home 3 cases of candles. And you know, well, great, what am I going to do with 3 cases of candles that I'm going to get every day for doing this work? I can't possibly burn that many candles. Can you pay me? The reason you want to get paid is the money is a little bit more universal. You can take that, you can go to the grocery store with it, you can buy new clothes with it, you can pay your rent, you can buy your car, your gas. And that's what the ATP is like. It's something a little bit more universal where we can take that energy from the ATP and spend it lots of different places in the cell. So when we're doing chemical work, what that means is we are pushing endergonic reactions in a direction that is not spontaneous. So you're going against the grid. You're having to push things uphill in there. Transport work is going to be where you pump substances across the membrane against the direction they spontaneously want to move. So things usually want to go from the higher concentration to the lower concentration so they can be spread out equally. It's kind of like with a little kid. If you give them a box of toys that's all in one corner, automatically they will fill the room with the mess in there. So that's like having where you're having to go along and constantly say to the kid, no, let's put the toys away. No, let's put the toys away. No, let's put the toys away. It's just going against nature in there. Mechanical work, that would be things like beating cilia, contracting skeletal muscle. And so with energy coupling, you use the exergonic process to drive an endergonic one. And this is showing it here using a hydroelectric system in here. So you're using the dam water to spin the turbine in there to get some energy. And you can have it be all done in one, or you can break it down into smaller sections. By breaking it down into smaller sections, you still get the same total at the end, but here you can break it up into smaller parts and say, okay, I'm going to take and budget this much to go here, this much to go here, this much to go here. And this is what the body will tend to do, is break it down into smaller sections so it can take smaller amounts to go in different places. And the way it will do this is with ATP. So let's go ahead and take a break for a couple minutes. So this ATP that we're going to use, we're going to talk a little bit about this right now. It's adenosine triphosphate, so it's going to have an adenine in there, and then it's going to have 3 phosphates attached to it, and it also has a ribose in there. What is significant is where these phosphates get attached on in there, it holds a lot of energy in that bond of holding on, especially that 3rd phosphate in there. So to take it where you hydrolyze it, you add water to it, the ATP adding water to get ADP, so it goes from triphosphate to diphosphate, and you pull off one of the phosphates, has a negative delta G of negative 7.3 or kilocalories per mole or 30.5 kilojoules per mole. Again, you don't really need to know that number for anything in here. Under standard conditions in there. So that's going to be how much energy that it's going to hold in that breaking and releasing that bond in there. That's under standard conditions. Under cellular conditions, it's actually even more efficient. It almost doubles in there. It's 78% greater. So it goes up to negative 13 kilocalories per mole in there. So what's useful in that is because most of the energy that is released in the cell, you're going to have this be greater than what most other molecules are going to be involved in. So it's going to, because of that difference in the energy in it, it's going to spontaneously choose to use the ATP for the reactions in there. So if you're going to have 680 kilocalories of energy being broken off from 686 from the glucose being broken down, you're going to need to form a lot of ATP from that at 13 kilocalories per mole. So that just gives you an idea of the rate of how many ATP you're going to get per that. What it comes out to be in a cell is usually 36 to 38 ATP per gram of glucose, or per glucose broken down. That's looking at a mole of glucose being broken down. So it just starts getting bigger in there. So how ATP performs work. ATP is going to harness the energy in there. The cell proteins harness the energy from the ATP in there, and it will be coupled with other endergonic reactions. Usually it's going to involve phosphorylation, so transferring of phosphates in there to transfer the energy. So this is showing what a big ATP looks like in there. This is simplifying the model down a little bit in there. And this is showing when you hydrolyze it, all you're doing is breaking this one bond in here and releasing some energy in the process of doing that. So some examples of ATP working with transport and mechanical energy in there. So if you want to transport these molecules here, ATP will change the shape of this protein that transports things and pull it up in here, change it out, spit it out on the other side in there. So ATP is used to help take that energy change shape and push the molecules up and out in there. It's used to help move the motor proteins for causing the vesicles to move along your cytoskeleton tract. So you're going to constantly be regenerating ATP in the body. So when you eat, that's what you're doing, is trying to regenerate that ATP. And it comes from the exergonic of the breakdown reactions. So that's when your body is breaking down carbohydrates, it's going to be doing that. So enzymes, they are going to play a role in this. They help to speed up the metabolic reactions by lowering the energy barriers in there. So sometimes you have a reaction that wants to occur spontaneously, but it's got this little hill of energy that it has to get over before it will do that. We refer to that as the activation energy in there, the energy required to contort the reactant molecules enough for the bonds to break in there. So you'll have a transition state in there where it has to go through. So this is actually supposed to be a smooth line, but I drew that in there, and I don't draw well with a mouse. So this shows it here where you've got the energy of the reactants. This transition state is actually a little bit higher, and then your products are way down here. So you have to put in a little bit of energy to get things going. So if you think about it, when you put briquettes in your barbecue in there, they've got lots of energy to cook your food. However, you don't just put them in the barbecue and put the food on and say, cook. You've got to put a little bit more energy into it in order to get that out. So that's when you put in the lighting the matches and the lighter fluid. You're putting in more energy to get those burning so that it can release it. So your lighter fluid and matches are helping you get over this. And we have to do the same thing a lot of times in the body. And what enzymes do is help you get over this little hump. So ways that enzymes will do this in there, you don't necessarily want to do that by heating things up. If you heat things up, you speed up all the reactions around it, so that wouldn't necessarily be a good option in there. But the enzyme doesn't change the delta G, so instead what it may do is maybe bend things, or it's going to move things a little bit closer so that it helps to do that. So you can think of it as the enzyme brings you the matches and the lighter fluid to get over there. It's the helper in the process in there. So it actually lowers the enzyme, the activation energy in there. So it's making the hump easier to get over. So with enzymes, you don't want them just going around and handing everybody lighter fluid and matches in there. We've got to control it somehow in there. So what they'll do is often come in multiple pieces in there. So you may have somebody that brings you the lighter, somebody that brings you the matches, and hopefully by doing this we don't give lighters and matches to little kids. It's kind of the idea and the process in there. So when we look at the parts, the substrate is going to be the reactant that the enzyme acts on. It would be like your briquettes. So when the enzyme binds to the substrate, you end up with an enzyme substrate complex in there. A lot of times enzymes are going to end in ASE ending. So if you want to break down maltose, you have the enzyme maltase that works with it. So the enzyme names are real similar to what they work on. So the enzyme has an active site in there, which is just a restricted region that's going to bind to the substrate. So the substrate goes into the active site here, and then you've got the enzyme substrate complex. And it's very specific, so you can't put something in that's a different shape in here. That enzyme is only going to work for a specific thing in there. So we say it has an induced fit. So as the substrate enters the site in there, the chemical groups cause the enzyme to change shape a little bit. So it just kind of hugs it a little bit more. Once it's in there, it says, oh yeah, I like having you in here. I'm just going to grab you in there. So when the substrate is held in the active site, it catalyzes the conversion of the product, and then the product departs the active site. So it will come in, it works on it, and lets it go out. So a single enzyme can act on thousands of substrate molecules per second. It acts on one, lets it go, another one comes in. So enzymes can actually end up controlling the rate of a reaction in there, because if you've got one enzyme, it can only go so fast. If you've got two enzymes, now you've got twice as many helpers in there. So the more enzyme you have, usually the faster the reaction rate can go. So a lot of times the enzymes can catalyze things both forward and backwards in there. So things it can do, it can bring the reactants together in proper orientation. So it may say, okay, I need you two things to work together. You're going to come in, and they come in like this, and the enzyme will twist them to fit like this. So it says, okay, you guys need help. We're going to make you right to help you go in there and there. It may stretch them towards the transition state form. So they may come in and it may just bend those bonds a little bit in there, putting a little more tension on them so they break a little bit easier. It can provide a microenvironment more conducive to the reaction itself. It may make a microenvironment that changes the pH a little bit in there. Sometimes they can directly participate in there where it will briefly covalently bond with one of the R groups and then unbond with it. But you always have the enzyme go back to what it was beforehand. So it's not actually consumed in the reaction. So if it bonds with it, it has to undo it in there real quick. So it may say, you know what, I'm going to do this part for you, but then I'm going to hand it back to you and it's yours. Keep going. So the rate of a reaction is partially the function of the initial concentration of the substrate. But at some point in there, the enzymes are going to become all saturated. All of the active sites are taken up, and no matter how much more substrate you put in there, it can only work so fast. So it's kind of like if I said to you, you need to read this chapter tonight, and I look and I go, oh, they did one, that was good, let's try more. If one is good, let's try more. And I give you 2, and then I give you 3 to read the next night, and 3 is all you can handle. There are so many hours in the day. Even if I give you 10 or 20 in there, you can only do so much in a time period. And so same thing with the enzyme. It can only do so much in a time period. So adding more of the substrate isn't necessarily going to indefinitely increase the rate. So this is just showing them come in here, or it comes in, lowers the activation energy, you change something, you get the products out, the enzyme's ready to go again. So how fast can you spin the cycle in there? So some other things that can influence it, temperature and pH. Being that it's a 3-dimensional structure, it's going to be sensitive to its environment. Every enzyme is going to have its optimum environment to work in. That's going to favor the most active shape. If you heat it up in there, what will usually happen is it may work faster for a little while, but you'll reach a threshold where it just says it's too hot, I can't keep doing this in there, and it will change it. Same thing with the pH in there. So what this represents is every enzyme is going to have this optimal temperature that it works most efficiently at. So for most enzymes in humans, the optimal temperature is going to be 37 degrees Celsius in there. So for a thermophilic bacteria, these are heat lovers. It may be 77 degrees in there. So if it's only 37 degrees, the enzyme's not even going to be active for that one. So the temperature would influence it. For that human one, if you raise the human's temperature up to 77 degrees Celsius, you'd be dead. So same thing with the pH in there. So you've got some enzymes in your stomach that are going to work optimally around a pH 2 in there. And so optional for trypsin in there. What you consume for food, though, you need the pepsin to break down, not the trypsin. So cofactors will also help in there. These are non-protein helpers for catalytic activity. A cofactor is going to be an inorganic molecule such as a metal or a mineral in there, where a coenzyme is going to be an organic cofactor in there. So that's the difference between them. So things like vitamins will act as coenzymes in there, or materials made from vitamins. So a lot of times when people say, well, you know, I just felt like I had more energy when I took vitamins. You didn't actually get any energy from the vitamins. What the vitamins did was help increase and make your enzymes all active that you had. You weren't getting enough of them before, and so what you did is you made enough of those cofactors for the enzymes to work in there. It wasn't the vitamins themselves giving you the energy in there. So anybody have any idea which vitamins are involved with most of our energy metabolism? Which group? The B vitamins in there. So most of the steps of metabolism are going to have a B vitamin as a coenzyme in there. What was that? Oh. So then there's going to also be things that are going to be inhibitors in there. A competitive inhibitor is going to mimic the substrate, so it's going to compete for the same spot that the substrate goes in. If that goes in there, the substrate can't go in there, and the enzyme doesn't work for it. So you can actually overcome those by increasing the concentration of the substrate so that you just have so many more of the substrate, the good substrate, competing for the space that the inhibitor doesn't really stand a chance of getting in there very often. Likewise, you could put in a whole lot more of the inhibitor so that the good substrate doesn't necessarily get in there. Non-competitive inhibitors are going to bind the enzyme in a place other than the active site, and then it alters the shape of the enzyme. So a lot of toxins and poisons are actually irreversible enzyme inhibitors in there. So they bind to it permanently in there. So sarin nerve gas is one. The pesticides DDT and parathion. A lot of antibiotics are also going to be inhibitors like that. So regulating the enzymes can help control metabolism in there. Allosteric regulation means the protein's going to function at one site in there, so it will be affected by binding of a regulatory molecule to a separate site. It may inhibit it or it may stimulate it. So most enzymes are going to be allosterically regulated, and it's going to have 2 or more subunits, each having their own active sites. So where we were looking at the picture before and it's saying, okay, you have to have this cofactor on there, usually it's going to involve there being multiple pieces and all of the pieces having their cofactors. The body likes to have a backup plan in case one form of regulation doesn't work. It regulates it at multiple levels in there so that if one little thing goes wrong, you don't just die instantly. So that's a good thing for keeping us alive. In terms of when we have to figure it out and learn it, you're just looking and you're going, really, can it be a little simpler in there? But that's why it's designed that way. So when you bind that activator to a regulatory site, it helps to stabilize the shape in there. And when it binds an inhibitor, it stabilizes the inactive form. So it's just stimulating it or inhibiting it by which helper you give it in there. So the fluctuation in the concentration of regulators then will influence the response of the activity in there. So with cooperativity, this is going to be the substrate causing an induced fit in one subunit can trigger the same favorable shape change in all the other subunits of the enzymes. So it can take one thing and say, you know what, this one is so important and we trust it so much, you're going to influence multiple subunits in there. And so you can also use that as a way to amplify the response in there. It can prime enzymes Hemoglobin in vertebrates is an example of cooperativity. Once you bind a little bit of oxygen, it's just going to increase your affinity for it. You're going to want more of it. So the first one comes in, it's the hardest one to bind, but then the others are even easier to bind on there. And in the body, that's actually helpful because if you relied on diffusion alone to get your oxygen in there, you just have 50-50 on both sides. You get 50-50 in your lungs, 50-50 in your bloodstream. But by increasing the affinity for it, it wants it more and more, and that's going to help to draw more out of your lungs into the bloodstream in there. So it's kind of a way of overcoming nature in there. So the allosteric regulators, we assume they're probably widespread, but they're really hard to isolate and characterize in there. So we know they're out there, we know they play a lot of roles in there. We've got a few examples, but we know there's places that these are working that we just haven't identified yet. So feedback inhibition. This is a way that you can shut down a metabolic pathway when it's made enough of something. So what it makes, the product at the end, as that starts to build up, it will start to shut down the metabolic pathway so you don't continue to make more of it that you don't need in there. So you can also help to control them with having specific locations for enzymes within the cells in there. So that is the end of Chapter 9.